Right. Well, good morning. Say good morning. All right. Good. Exactly. Well, we got to compete with those folks across the way here. We're all charged up. You guys should be really all charged up. At least I am. I thought we had a fabulous day one, and we're looking forward to a fabulous day two. I love the energy and the cross-pollination going across the various fields and disciplines, diverse sectors of this ecosystem that are in this room. And I anticipate the same thing will happen today as we continue to go from head to toe. As a cardiologist and director of the National Musculoskeletal Institute, I must admit my bias, despite my being ecumenical in this meeting, that we have some great topics today in terms of neurological, the brain, and as well as the cardiovascular system and the blood stream. So we look forward to some great dialogue today. And without any further ado, we'll turn it over to Dr. Jones.
Good morning, everyone. My name is Candace Kirk. I'm a program officer at the National Institute of Aging, and I will be co-moderating
My name
this
is Eric
session
Rose. I'm
with
speaking
Dave
this
Owens,
afternoon.
the acting deputy director
I have a
of the
revised slide
Division
deck
at our National Institute
that I just gave them up there, and they
Neurological
told me to bring them
Disorders
to
and
you
Strokes,
as well.
also at
Okay.
NIH.
So it's
We will
on there.
begin the session with two presentations, one with the state of clinical science and the other with the state of product development, each including an illustrated case study. If time permits, then we'll follow up with a question or two from the audience. And then we'll follow with remarks from panelists here at the table, discussion of key questions, pretty much like we did yesterday.
What session are you in?
Sometime after
Cardiovascular.
audience.
Do you know what time it starts? Yeah, 12:30.
On time, as much as possible. Our first speaker, Dr. Robert Mays, is the Vice President of Regenerative Medicine and the Head of Neuroscience Program at AfroSys Inc. He will present on the state of clinical science and neurology. To help keep us on time, staff in the front of the row will hold up a yellow card.
This PDF?
One
Yeah.
that remains in a red card when time has expired.
Good morning, everyone. Thanks for coming so early, and thanks to Amy and the chairs for inviting me to present this meeting. So, we're a publicly traded company, so I have
Okay.
to do that.
Great.
And
Thank you. Thank you.
You have some disclosures, so take this into consideration as you guys need to talk about our side of the product. So very quickly, going to go through the landscape of neurology relating to stem cells, introduce you to our proprietary multi-stem product.
No, it's a PDF file.
Preclinical data that's relevant
So it can work.
to our translational program in the clinic and our upcoming phase 3 study. So according to the Alliance for Regenerative Medicine, or as of the end of Q3 this year, there were almost 1,000 clinical trials, phase 1, phase 2, phase 3 across all of the designations of the regenerative medicine landscape. The majority of these are in the oncology space, obviously the CAR T programs driving a lot of that, but in the central nervous system and in neurology, there's roughly 58 different trials taking place currently with regenerative medicine products. A very important part of, I think, how you're going to set up a think about your clinical program has to come from the idea of understanding what are your cells doing, regardless of whether you're taking adult tissues allogeneically, whether you're using autologous products from bone marrow or cord blood, or whether you're chemically or gene modifying cells to reach your final product, to expand and then either differentiate or manipulate in a secondary way. What you make, where you place it, how you place it, when you place it, how much of it are you placing are all critical parameters that need to be contemplated. So if you're going to think about replacing, for example, neurons in the brain after a stroke, it certainly doesn't seem to make logical sense that you would want to do an IV administration of a fat-derived cell, because that just doesn't work. So if you're going to do an intrathecal administration of a cell type that become a neuron or is a neuronal precursor, now you're talking about a logical clinical translational step. So this slide, the next slide, basically covers the landscape of some of the big indications in the neurology space, which important clinical trials are currently being done. I took the data from these two slides off of clinicaltrials.gov went through clinical trials. I got one of the as a little bit of adverse press yesterday. I did go through and only pick out trials from organizations or medical centers that I know are responsible, thoughtful programs and with trialists that I know. So you can see there's a lot of different cell types, a lot of different routes of administration. IT being equal, IV obviously intravenous. So just to highlight some of the trials going on. Our product is the multi-stem product. It's an adult adherent proprietary cell that is based on the original multi-protocol progenitor cell. So these cells can be used off the shelf without matching. I'll talk about how we believe the cells work in the next slide. We understand that these cells will do different things depending on where you give them and how you give them. So we systemic administration, we've got local implantation. We see the cells do different things depending on that context. And with guidance from the FDA, we've been monitoring these cells from a clinical standpoint. We have greater than 7 years of stability frozen product. We know that from one single bone marrow donation, we can make literally millions of doses from one donor. So what we've done over the course of the last 10 years since acquiring the intellectual property around this cell type is spend a lot of time and a lot of in vitro and in vivo models to understand exactly what we believe works to better understand what the cells are doing. So basically, these cells are not going in and not becoming tissue. They're not becoming neurons when we give them the stroke. They're not becoming cardiomyocytes when we give them the acute myocardial infarction sense. They do drive the body's innate cellular regeneration more strongly. The major mechanism through which these cells work is via immunomodulation and changing the balance of the inflammatory response after stroke or after injury. So this slide is up there for two reasons. One, to remind me to thank the NIH. Almost all of the work in the neuro space that me and my group have worked on over the course of the last 10 years has been supplemented by the National Institutes of Health and grants and or from other agencies. The second point is we have identified that there seems to be a conserved mechanism through which the cells are working in acute neurologic injuries, stroke, traumatic brain injury, hypoxic ischemic injury, concussion. I won't be showing you that is spinal cord injury. And that maybe the same conserved mechanism might apply to non-neurologic injuries like trauma, acute severe trauma as well. So I'm going to be focusing on stroke for the remainder of the talk. But there's a lot of data that is conserved between our stroke program and other neurologic injuries. Stroke is a very important indication. Leading cause of disability, the biggest drain on our healthcare system. In 2018, it's believed that there will be 18 million strokes worldwide. 6 million will die, 6 million will be left with significant deficit after the stroke. There are only a couple of curative or reparative ways to treat stroke, TPA, mechanical perfusion. They have to be, patients have to receive the treatment within the first 6 hours to really have any chance for benefit. So this really limits the number of patients that can benefit from these modalities. So severe right-hand turn here. So about 10 years ago, people in the stroke neurology space started to recognize that the spleen was critically involved in post-stroke immunodepression and inflammation. This slide basically highlights, if you look along the top, this is Floro J. Greenstein, a sham-injured animal, a stroke-injured animal, and a stroke-injured animal who had its spleen removed 2 weeks prior to inducing the stroke. You can see that the bright lime green color goes away in this splenectomized animal. There's far less injury to the brain in this splenectomized animal. From a different lab, Selena Hoffner and Patricia Hearn, when they were up there working with health sciences, on the left-hand side, A is the spleen of a sham-injured mouse, B is the spleen of a stroke-injured mouse, 96 hours after the surgery. So you can see, and also not to scale the fineness of C and D. So obviously the peripheral immune system is involved in the post-stroke pathophysiology. So this is work that we did in collaboration with Sean Savage at the University of Texas Houston, who's sitting right over there. And so basically, the spleens of two stroke-injured animals, a multi-stem treated animal, and a vehicle treated animal. You can see that the cell treatment stabilizes the splenic atrophy Sean has gone on to publish a couple of papers demonstrating the same phenomenon takes place in humans as well. So on the left-hand side, the blue dotted line is normal controls. The red shows hour by hour sort of breakdown of stroke survivors. So you get a decrease in splenic mass followed by a rebound. We also see the spleen is involved in some of the cytokines that are known to be inflammatory post-stroke. So on the left, it's animals with spleens. On the right, it's animals that have been splenectomized. So in IL-6, you see IL-6 levels go up over the course of the first couple of days after inducing a stroke. This is knocked down or statistically taken away by our cells. But the level of IL-6 is completely plummeted when you splenectomize animals 2 weeks prior to inducing a stroke. We also see change in the phenotype of T-cell populations in vehicle versus multi-stem treated stroke animals. So Tregs are actually going up in multi-stem treated animals while we're knocking down CD4 and CD8 plasma cells. So with some of that data in our back pocket and some safety studies that we did in accordance with the FDA, we published earlier this year our phase 2 double-blind placebo-controlled stroke study using our cells. Basically, I'm going to show you just a couple pieces of data. That the original trial design was treating patients 24 to 36 hours after the onset of stroke, 1.2 billion cells delivered IV, safety, multiple clinical scales, and then some of the exploratory secondary endpoints to support our translational idea. During the trial, we needed to make a couple of protocol amendments based on some logistic issues. So we had to extend the window from 24 to 36 out of 24 to 48. And we also started to include patients who got both TPA and mechanical reperfusion. And that caused, we believe, some problems with the data as well. There are all 33 centers that participated in the trial. It was a well-balanced trial from a demographic standpoint. There weren't any imbalances throughout the safety profile.
How does that sound to you?
There weren't any serious adverse events or anything that stood out from a safety standpoint. We did miss our primary endpoint. We did see some of these exploratory and secondary endpoints trending in the right direction. When I go to this next slide, we did look at patient outcomes from day 90 and also at day 365 for animal data. You can see looking down the entirety of the trial. We did reach statistical significance at 9 days, but we looked at the entire population by day 365. We did reach statistical significance in our excellent outcome, which is basically a patient getting entirely back to normal. Again, these were severely injured patients. And it gets better as you go all the way down, the final point being the original trial design. So you can see roughly 3 out of every 10 patients was getting back to normal in the cell treatment. We also, from an exploratory standpoint, we modulated the decrease in the CD3 positive T-cell populations, and we affected the inflammatory cytokines exactly we had seen in the animals. And you can see bars and lines, we saw almost immediate knockback of the inflammatory cytokines. We also impacted the time in the ICU and total time in the hospital in the cell-treated versus the placebo treatment as well. So with that data, we have had our end of phase 2 meeting. We've applied for and received RMAT designation, special protocol assessment, and fast track designation from the FDA, as well as an EMA positive opinion. This trial will be commencing in 2018, and we're going to look at the spleen and do some MRI imaging as surrogate exploratory endpoints to see and continue to build off of our hypothesis. So I'd just like to acknowledge and thank the clinical teams, the patients and their families, and answer any questions if there's time remaining. Thank you very much.
Thank you, Robert. We have just a couple minutes for any quick questions.
Hi. This might be a little off topic, but I'm just wondering how you do informed consent in a trial like this. Nobody knows they're going to have a stroke, so who actually does the informed consent? So
we have it built into our informed consent. It's either the patient, unless they're aphasic, and then it has to be a family member or legal. It's no problem.
And please state your name and affiliation.
Hi, Josh Hare, University of Miami. Congratulations on a great study. You alluded to a conserved mechanism of action, but I wasn't clear what the conserved mechanism of action is.
Yes, so what we believe is that when there is a severe trauma or a severe engagement of the peripheral immune system, like you would see after a stroke or a traumatic brain injury, a lower body trauma that basically sends out a 5-alarm fire to the peripheral immune system. The cells leave the peripheral organs, the spleen, the lymph nodes, the thymus, and you end up being immunodepleted. Actually, it's well known there's something called SIDS, CNS-induced immunodepression syndrome. In most stroke patients, I find it to be an amazing set of data. There was a German group that published this roughly 8 to 10 years ago. If you survive the first 24 to 48 hours after a stroke, a TBI, a spinal cord injury, and now more recently, data from trauma has come in that shows this as well, you're more likely to die of a secondary infection. Pneumonia, ARDS, sepsis, some immunologically imbalanced manifestation than the original insult. So we believe our cells go in, understand the context of the 5-alarm fire, and actually keep the immune system rebalanced, keep cells from leaving the peripheral immune system and going to the site of injury, and therefore leaving the patients immunodepressed.
One more question.
Eduardo Marban, Cedars-Sinai. The fact that you missed your primary endpoint, I think, is less reflective of the bioactivity of your cells, which is seemingly amply demonstrated by the other data than it is by a problem that I think plagues the field, which is that in a product that has multiple mechanisms of action, sometimes years, many years before you actually complete the trial, you have to look into a crystal ball and say which of those many mechanisms of action is going to be the best biomarker. And then when you go to multi-site trials, it differs a lot than for these carefully curated single-centered studies where you can have absolute control over phenotyping. So I'm just saying perhaps we need to rethink traditional clinical trial design in this field.
I think that's a very good point, and I appreciate that comment.
Thank you, Robert.
Thank you.
Our next speaker is Dr. Lorenz Studer, who is the director of the Center for Stem Cell Biology at Memorial Sloan Kettering Cancer Center, and he'll present on the state of product development.
Starting at the end here? No, we need to. Can you start? So just take the time when you set this up to thank obviously for the invitation. I really enjoyed yesterday's day and I look forward to all the discussions today. Now, we just heard, I think, from Robert, obviously, the number of trials going on in the neurology case using stem cells. And obviously, in most of those cases, these are stem cells that not necessarily are designed to actually replace the specific neurons lost in the disease. To really achieve that effectively, most people think currently the most effective way to do that is actually using pluripotent cells. And in the area of Parkinson's disease, where I will primarily focus on, there are actually a number of efforts ongoing across the globe, where indeed both autologous and HLA-matched cell lines are being pursued for use in Parkinson's trials, as well as non-matched efforts using ES cells or non-matched iPS cells. And the approach we would like to pursue long-term, also alluded to yesterday, which would be a universal genetically engineered cell line, which we think could combine some of the advantages of having an off-the-shelf product, relatively easy and cost-effective to manufacture, but not needing immunosuppression, particularly for the CNS. Now, for all those pluripotent sources, the key question, and that's really what was the work of the last 10 years, is how do we make the right cell type? If you want to have a cell replacement therapy, we better have the right cell type. And by now we have very good techniques to just simply have access to the nervous system by using just, for example, in this case, 2 small molecules that drive pluripotent cells into CNS lineages. And we just published very recently an updated technology that is used in a fully defined media with, again, very defined factors. And you can access not only the central nervous system precursors, but you can make peripheral neurons, you can make plaque-derived sensory structures, and you can even make precursors of the surface ectoderm using that in a very effective way. And the point that I bring that up is really that in the pluripotent space, I think we are now at a stage where we can recreate large amounts of the lineage tree quite effectively. In my own lab, we can make about 40 cell types routinely. And so we are now kind of at the verge where we exactly need to have this interface to see what for each of those cell types, even if we can produce them in a reasonable manner, what really can go forward and how do we get them effectively into the clinic. And I will focus on Parkinson's today, but we are also very interested in oligodendrocytes and, for example, enteric neurons in gastrointestinal disorders. Now, for Parkinson's disease, it's quite obvious why that's an interesting indication, because the key motor symptoms of the disease are caused by the loss of a discrete population. You have about 500,000 dopamine neurons in substantia nigra. If half of those are gone, you develop the motor symptoms. And those are cells born about 8 weeks after you are conceived. They are never replaced throughout life. So the only way we think we can get them back is recreate them in addition, put them back. And so we have to be assured, also make sure that we are understanding what we could offer the patient. We're not going to cure Parkinson's disease because there are also motor symptoms that might not be affected by such an approach. But the dream is to basically have a cure, at least permanently, of the motor symptoms of the disease. And there's clearly a need for that because obviously Parkinson's disease is the very second most common neurodegenerative disorders, and the current medication is still suboptimal today. But it's worth mentioning that there are a number of therapeutic strategies, including DBS, that can be quite effective. So in this case, it's not that we need to be just safe and effective, but we also need to have a product that's actually competitive with what's out in the market there. Now, the idea of replacing cells has been around for a long time, and we're again kind of in a unique situation where we actually can learn from what was done in the 90s using fetal tissue. Because it was shown if you take fetal tissue, put it into the brain, you can get dopamine neuron precursors from the midbrain to survive and to produce dopamine, such as, for example, by PET imaging. And this was done in over 400 patients worldwide, so we have quite a bit of clinical data as well. And while the results were variable, we understand now much better, for example, with regard to the patient selection, what's the right patient to choose. And obviously, we want to go forward with a much more standardized product than what you could do ever with fetal tissue. But I think most excitingly, in this case, is we actually have very, very long-term data from some of the patients, 15, 20 years out. And it shows that actually, at least in a subset of the patients, you can get a result where a patient at this very late stage, theoretically, of the disease, they're off L-DOPA medication, and they have dopamine levels in the striatum that are within the normal range. So this kind of proves that the therapy can work, but the question is, can we make it a consistent therapy? And so again, this lists some of the advantages that we hope to get really with a stem cell-based approach, not only replace dopamine, but potentially replace the synaptic network, and therefore maybe slow down the network degeneration itself. But practically, really, the therapy, what it would be is a one-time treatment, where in a one-time treatment, you would want to permanently cure the motor symptoms of the disease. Now, to really make that possible, it was again important to have about 10 years of developmental biology work to come up with a system where we can not only make dopamine neurons that look okay in a dish, but that function in a mouse, rat, and a monkey model of Parkinson's disease. And for example, if you look at those pluripotent derived dopamine neurons from a human in a monkey brain, you can appreciate that the morphology and some of the features is actually indistinguishable from the endogenous dopamine cells in the brain. And we can get survival of about 100,000 of those dopamine cells routinely in a monkey brain, and that number is about the same number that was found in autopsies of patients that did particularly well using those fetal grafts. So we are clearly in a therapeutic range. We have done a number of assays now to really measure basically improvement. You have these classic rotation assays. This is a sped up movie, but you can very easily see basically a diseased animal rotates around its own axis and it recovers once you replace dopamine on the side where dopamine is lost. Or you can have these slightly more sophisticated assays where you see basically skilled four-limb use. You see the animal on the right side that basically can first not use the left paw, but then starts using the left paw again after a graft. And that's not because the animal was paralyzed. It cannot really initiate the movement. So it's very similar to what you would see in a Parkinson's patient when they cannot properly initiate movement. What's also, I think, unique in this approach is we know it's not only a correlation. You put the cells in, the animal gets better, but you can actually show causation because you can use optogenetic techniques where you put the graft in, but then you actually shut it off. You shut off only the grafted cells, and all the behavioral improvement goes away. So you know it's activity-dependent dopamine release that really causes the improvement, and we can even show that these cells actually synaptically integrate and modulate the correct medium spiny neurons in the striatum, which is what normal dopamine neurons do. That's quite beautiful work actually from Suzhou Chang's lab that confirms that with another technique. Now again, to get that product into the clinic, it was then again important obviously to have the resources to do that and really go through all the steps to really make that not just a lab product, but a product we can use to move forward. And we were fortunate to have basically a state-of-the-art GMP effort together with Isabelle Rivière, Michel Sadelin, Sloan Kettering, to really help us guide through that process. And actually now being able, together with a team led by, again, Isabelle and Mark, to really produce those cells. And we actually have now already, again, I skipped about 3 or 4 years of work of how we adapted it, but we were able, by the end of last year, in fact, produce about the equivalent, what we think is about 1,000 doses of these human products, about 10 billion dopamine neurons that can be frozen down, and that follow all our pre-specified release criteria. And we did that actually 4 times with independent runs to show that every single run would meet or exceed those pre-specified criteria to make sure, again, that we have really a robust manufacturing process. Again, one key concern in the brain was, can we freeze young neurons? And that seemed to be the case, that we can freeze thaw the cells, and also importantly, when we thaw the cells, show that the stability remains actually pretty good over time. So when you get the cells into the surgery suite, you're not worried that they're going to die off immediately, but they are stable for about 24 hours. They are also stable, obviously, when they are cryopreserved, so you have to have a stability testing program because the cells that we're going to use, hopefully, in our phase 1 study are already produced. Again, I'm not going to go through all the various markers. I don't think I have time, but the point is that you have to obviously select those very carefully, because when you put the cells in, they're not fully mature. So for example, tyrosine hydroxylase, a classic dopamine marker, is clearly not going to be in all the cells. But we have other markers, such as, for example, FOXA2, which is a precursor marker, where we can show that at least based on those markers, the product is pretty highly enriched. So it's, in this case, this specific around 99% pure for those FOXA2 marker. Then obviously you have to show the absence of pluripotent marker, and we have a number of different assays, both gene expression, flow-based, and high-content screening-based, to show that we cannot really detect any pluripotent cells. But there you get into the question, obviously, what's the sensitivity of your assay? Now, importantly, is to show that if you have those frozen cells, do they still work in vivo? And in fact, we can now take a frozen vial, thaw the cells, directly inject them, in this case, into a rat model of PD, and get this very nice survival and re-innervation of the striatum. And importantly, we get correlative improvement functional in those relevant PD model that we are using. And again, we think that the function is really due primarily not just to the survival of the cell body, but to the fibers. So it's very important to show that if you go away from the graft, that you still see a lot of human-specific fibers that re-innervate the striatum, and that many of those are actually dopaminergic. So that's, again, a very important feature that's going to be particularly important in the larger human brain. Now, again, we went now through the various steps of pre and pre-IND, and we were kind of happy to hear that at least most of the criteria with CMC seem to be acceptable of the way these cells were produced, because obviously we already produced them, so they're better in good shape at this point. But there are still some additional areas that we're now just covering and actually just finalizing those studies. Obviously, we wanted to have basically device compatibility is an important issue. And we also need to show further the safety. And what's particularly interesting now in such a product is because you have really shelved all those cells, you can now take the exact same cells. So the exact same cells we're going to use in the trial went again into more than 400 mice for the classic tumorigenicity, biodistribution, and toxicity studies, and also for the efficacy studies, and actually a short-term monkey study where we use the exact same device we're going to use in the patients. And so again, that allows you to do that in the pluripotent cells because you can produce enough of a given batch. And this is then just in a nutshell some of the setups for the clinical trial. You might see on the right side, again, there's actually a small move if you look carefully, where we use the same MRI-compatible device to inject. In this case, it's actually a drug that's injected into the striatum. So you can follow exactly where it's injected. And it's particularly valuable to understand also the safety of the surgical procedure. Because if you talk to a stereotactic surgeon, they tell you that each needle track you put in the brain, to, for example, place the cells, has a certain risk of causing bleeding. And what we would like to propose is a one-time treatment that you could treat both sides of the patient's brain in one session, so you don't have to come back. You don't get any sensitization immunologically. And for that, it's obviously very helpful that you can monitor very carefully, avoid blood vessels, and even if you had a complication, you would immediately see it. Now we plan for 10 patients in this case, and there are many, many criteria, obviously inclusion-exclusion criteria, but one important one with regard to potential efficacy is that the patients have to still have a defined levodopa response. That was the most predictive marker in the fetal grafting studies. And then we plan to have 2 doses, a lower and a high dose. And again, in this case, it's actually quite interesting because we actually kind of know the dose, what we need. Because we have, again, from the fetal studies, we have autopsies, and we know that our cells have about a similar potency on a per dopamine basis. And so we can tune that to get the number of cells injected to give us the appropriate survival. These patients need to have immunosuppression for up to 12 months. So we know that transient immunosuppression is enough to get long-term survival, but we still think we want to use transient immunosuppression. Again, that's a discussion by itself, and it's going to be a single-site study. And again, this just gives you a flavor of the timeline, and I put that there again also because up to now, pretty much everything we did is in an academic effort with funding from the state, New York State, but actually we just started basically last year, a company, Blue Rock Therapeutics, so I have to start putting the slides up as well, because I'm a co-founder of this company, and we hope again for that money we have now the resources to move that product forward through the clinical development, and also obviously hopefully bring other products along the line, along the tree that I showed you before. Now, one point I want to make, a last point really, is that we try to do that also in an effort across the globe to kind of harmonize what we think is the safe way and the efficient way forward. So we formed an academic effort, it's called GForce PD, Global Force PD, which combines all the major academic centers across the globe that try to do that using either embryonic or iPS-derived cell types, and we have multiple meetings to really discuss things like clinical trial design and so forth. And we actually just came out with a new kind of conclusion from our last meeting that specifically discussed clinical trial design. I think that's going to be quite useful for us to know that we all agree what might be a reasonable approach, but also for anyone else that they get guidelines on really how to move forward in a reasonable manner. And last, I just want to acknowledge people. It's obviously a major consortium that makes that possible. I just want to highlight particularly Mark Tomishima, Stefan Irion. Mark was the guy who really was responsible for the manufacturing in large parts, and Vivian, who is basically the co-PI on the project and was important for all the preclinical animal studies and for also the surgical design. So I'm going to stop here and hope we still have time maybe for at least 1 or 2 questions.
Thank you, Lorenz. We've got time for maybe 1 quick question. I do apologize, but we'll have to move to our panel discussions. You'll have another opportunity also to ask then.
What's the immunosuppression regimen that'll be required?
So mostly it's just tacrolimus, short-term methylprednisone, and there's a question that we actually have MMP in addition to that. So, and actually basiliximab is also added.
And one more, name, affiliation.
Yes, Barry Baumel from the University of Miami. I wonder if you would just speak a little bit more about dosage selection, because my understanding is that the wrong dosage, you end up with abnormal movements in these Parkinson's patients.
Yeah, we actually don't think that's completely true. That has been interpreted in some of the fetal studies like that, but there's no clear dose correlation with the development of graft-induced dyskinesia. What does correlate is pre-existing levodopa dyskinesia. The patient already has, in response to the drug, dyskinesia before he gets the treatment or she gets the treatment, then very, very high risk. And with regard to the cell product, what seems to contribute is particularly contamination with a serotonergic cell that seems to be able to take up L-dopa, store it, but inappropriately release it. Fetal cells, they had nearly as many serotonergic neurons and dopaminergic neurons the way they were dissected. In the pluripotent product, we don't really have pretty much any serotonergic cells. So we think we can eliminate that point. Now, it still doesn't eliminate the question of dosing, and that's obviously important. We know from animal studies that you have a range, you can about 5 or 10 times increase the number of dopaminergic neurons without seeing much of a side effect. We wouldn't want to go that high in patients, but so it means dopaminergic neurons have some buffering capacity, which seems to be at the synaptic level, the dopamine transporter. So we're not so worried about overdosing, but clearly you have to be within a reasonable range. And with regard to having the effective dose, again, we rely on what we learned from the fetal grafts. Now, obviously, it could be different with the pluripotent-derived ones, but it's at least a good estimate to start off.
Thank you.
Thank you. Fantastic questions. Thank you, Lorenz. David will introduce our panelists.
Okay, so, excuse me. So we're going to do, as we did yesterday, run through some key questions. The first question, I believe, are we going to project them? Well, anyways, we'll project them. But so the first question we're going to have all the panelists address, give a few minutes on. Each of them will give a few minutes on it. So what we'll do is we'll start with Sean Savitz, who is professor and the Frank M. Yatsu Chair in the Department of Neurology at the University of Texas Health Sciences in Houston.
Should I go up there or stay here? Really, thank you for the opportunity to participate in this wonderful conference and this session. To me, what's really wonderful about this conference is you've just heard from two speakers that illustrate two major paradigms of how cell-based therapies are currently being investigated for different neurological disorders. One is clearly a cell replacement strategy, and the other is a very different strategy where the objective is not cell replacement, but more, I'm going to say, is immunomodulatory approach. And that's really a new paradigm, I think, for many different neurological disorders to think about actually that type of therapeutic approach and fundamentally what the treatment options are in this respect. So I think overall, for our panel discussion, some thoughts that came up with regards to this question is that for many different cell types that we're looking at for neurological disorders, the intended goal actually may not be engraftment or cell integration. Intended goal may be that for some disorders, but it may be something quite different for other disorders. For the kinds of conditions from Dr. May's discussion, they're far more directly related, the principal mechanisms more related to immunomodulatory and paracrine effects. And that may be involving the release of biological factors. Exosomes and microparticles is very big right now, as we've already heard from yesterday's talks. There are cell-to-cell interactions between the exogenously applied cell and the host cells. And there's a lot of work that shows a convergence that within the brain, some of the principal cell types that mediate inflammation, such as microglia, may be actually some of those targets. So there may be immunomodulation that's going on by cell-based therapies. Ultimately, in the end, through mechanisms we don't fully understand, the cell types within the brain that carry that inflammatory signaling process get impacted. And microglia may be one of the ultimate targets in that regard. To me, the three main areas for neurological disorders to think about from these discussions in many different ways is the following: that the post-injury environment for acute CNS disorders such as stroke, TBI, spinal cord injury, really are unleashing a cascade of well-orchestrated immune and inflammatory responses that can be therapeutically modified to enhance recovery by cell-based therapies. IV multistem, IV administration of multistem, I think exemplifies this approach to modulate the inflammatory response. This is likely a time-sensitive therapeutic window. It probably depends upon the right targets emerging at a particular time, and then those targets disappear. For the subacute to chronic period after acute neurological injuries, I think a big question for us all is, can cell-based therapies amplify endogenous recovery processes? And I hope we have some discussion more around that. And then for degenerative disorders, again, you just heard from a wonderful strategy for cell replacement with regards to degenerative disorders. But for immunomodulatory and paracrine mechanisms, there may also be a role for degenerative disorders. Will immunomodulation and these types of mechanisms be effective to perhaps slow down or even arrest progression of neurodegenerative disease? So to advance the fields and thinking about this overall, a few points to think about overall, that it's really important for us to think about linking the intended mechanisms and translating that to in vivo outcomes. And I think a lot of focus that we've had on looking at surface markers and differentiation capacity of MSCs is an old paradigm that may not really apply to a lot of the work that we're talking about here. It's very much important to understand from an MSC biology standpoint, but I would much prefer to see us move in the direction of looking at potency of the intended effects. So if that's immunomodulation, then we should be looking at potency assays for immunomodulatory effects that are linked to the outcomes that are desired in the brain. There's already very intriguing work in the traumatic brain injury literature looking at that, how MSCs modulate immune cells and how that's linked to what they're doing in the brain to reduce, let's say, blood-brain barrier damage and reduction of cerebral edema. I think we also need a far greater understanding of the intricate connections between the immune system's responses and the targets within the brain for cell-based therapies. If that's a way for me to think about how to advance the field, we need a far greater understanding of how cell-based therapies could be impacting the spleen and how that actually translates to positive outcomes within the brain. And there's also the concept of chronic inflammation playing a contributory, very prominent role in the progression of neurodegenerative disorders. This may be an excellent target for cell-based therapies, given their immunomodulatory effects. And then just a few more points to think about for advancing the field, that I hope that we can put more attention and effort into developing better and safer imaging capabilities so that we can image our targets within the brain. For example, PET imaging for microglia seems like, to me, a great example of where this could go. And I hope that there are other types of technologies like this that will be developed over time. And lastly, we haven't really gotten into this topic, but it was mentioned yesterday that multiple injection paradigm may be far more effective than single injections too. So I hope, again, in the field that once we get through our studies of doing single IV injections or single injections in general, that for some appropriate conditions that we move to the serial injection paradigm in the future. And lastly, I just wanted to mention that there are consortium that have been developed for different neurological diseases. Stroke, I'm just bringing up as an example, but there's partnerships coming together between leaders in academia, industry, coming together with the FDA and the NIH. And I think that these types of consortium are critical to be able to advance the field for various different neurological disorders with regards to the development of cell-based therapies. Thank you.
Great. Thanks, Sean. So now we'll move over to Su-Chun Zhang, who's a professor of neuroscience and neurology at the University of Wisconsin-Madison.
Thank you, Dave. It's been very informative for me to attend this meeting. I think as alluded by a lot of speakers, for most neurological conditions, whether they are degenerative disorders or traumas, we actually have relatively limited effective treatments. And so cell therapy is one potential option, whether going through immunological modulation, cell replacement. I think one of the major issues in cell therapy field is how cell therapy works. And I think that was brought up yesterday and actually also this morning. For example, in Parkinson's disease, as Dr. Studer pointed out, the reconstruction of the neural circuits, at least partially, is critical for achieving the therapeutic effect. And that has been done by the current tools like optogenetics, chemogenetics, and some of the related tools. And I think for many other neurological conditions, it may not be that clear. For example, for cell transplantation into the brain of stroke, spinal cord injury, and so on and so forth, we may say that they are achieved through modulation of immunological condition, trophic support, myelination, and so on and so forth. But I think nowadays we have tools like optogenetics, chemogenetics, that will help us define how the cells therapy works, which will actually help move the field forward and may potentially also inform what kind of cells, how do we put the cells to it. And so that comes to the technical issue, first thing, what cell types we are going to use. I think the brain and spinal cord is wired in such a precise way. Often we require a very specific type of cells, in particular neurons. And I think the good news is that we actually, a lot of labs have worked out, and we are now able to guide the stem cell to many different kinds of neurons and glia cells. And as pointed out, that for this reconstruction of the neural circuit, actually we need a very specialized neuronal types or glia types. For example, for Parkinson's disease, we need midbrain type of dopaminergic neuron. Not just that, actually, we need a specialized kind of midbrain dopaminergic neuron in order to achieve the reformation of the circuits. And so I think for that part, despite that we have achieved a substantial area of cell types, but still I think we need to work out for the other cell types that are specific to other neurological conditions. For example, if we need specific cell types for dealing with pain, dealing with spasm, dealing with autonomic nerve function, and so on and so forth. Because the nervous system, the cell therapy is to kind of reform the neural circuit. Usually, when we use the cells for transplantation, also becomes an important issue. Because during development, usually the immature cell or progenitor cell, they somehow are able to do it. After they become mature, and so in the mature nervous system after injury, they somehow are not able to do it. So identifying the right stage of the cells is also important. I think nowadays it's largely worked out that the neuroprogenitor cells, which are committed to certain faith, and they are generally appropriate for cell transplantation to achieve the goal. And now when it comes down to preclinical and clinical operation, it's also important to have some kind of objective outcome measure besides behavioral and clinical testing. For example, in Parkinson's disease, we have a PET scanning to monitor the transplanted cells, whether they are there and whether they are functional. And that actually can inform, whether how the cell therapy works, and that can come back to inform whether the cells we generated and put into the patient is the right one or not. So we can discuss more about that. I will stop here.
Great, thanks, Yu-Chun. So Tom, we'll get back to you in a minute. So next we'll have Scott Berger, who is principal at Advanced Cell and Gene Therapy, LLC.
Oh, thank you very much. Some very interesting discussions yesterday and this morning so far. In terms of scientific, technical challenges, I want to reiterate some of the points that were brought out yesterday in terms of the need for raw materials of an appropriate quality, quality grade, quality level, particularly since in manufacturing many of these products, we are using raw materials that are not commonly available GMP or as pharmaceuticals, and oftentimes we're having to qualify up research grade raw materials or research use only raw materials. So for the last several years, I and some colleagues on the CMC side of cell therapy have been trying to push manufacturers to move more towards either GMP or, let us say, near GMP quality reagents, particularly cytokines and growth factors and other supplements that are absolutely critical raw materials, but that are commonly available only as research use only. Let's see, a couple of other thoughts. Are we coming back to the scale-up, scale-out challenge questions later on, or should I tackle that now?
Yeah, well, I think we'll address it again.
Okay, then I'll touch on only a couple of other points right now, one being the need for analytical standardization, and for that matter, analytical standards. There was some mention yesterday of some recent NIST-sponsored workshops on cell counting and viability measurement and on flow cytometry. These are absolutely fundamental tools and are not at all simple to standardize. Silly as that may sound, something as fundamental as counting cells is really quite a challenge to do in a manner that is accurate, precise, and consistent across multiple centers around the world. So refining our fundamental analytical tools and standardizing them is absolutely essential. Also because we're generally manufacturing more than one kind of, let's say, MSC. There isn't a cellular standard that we can turn to, and probably won't be for quite a while. So if we can't standardize on this is what everyone agrees an MSC should be and how to measure it, we need to standardize on the how to measure it side of it, so that we can at the very least be using tools and analytical strategies that are consistent. Let's see, last off, I'll mention processing technologies. This is a particular challenge since oftentimes the processing methods and processing technology that we use in early stage R&D studies really has to undergo a substantial change in order to be suitable for GMP manufacturing, large-scale manufacturing, and that refers to everything from cell selection to, I think, especially ex vivo cell expansion, particularly for adherent cells. I think we'll talk about that a little bit more when we come to scale up and scale out. So, better raw materials, standardized analytical methods, and better cell expansion technology. So if we could have more of that, please.
Great, thanks, Scott. So we'll move on now to Ilya Sigesh, who's the head of stem cell research at the National Center Advancing Translational Sciences, or NCATS, here at the NIH. Ilya?
Okay, thanks very much for this opportunity. I had some slides, to basically reiterate some of the important points we have already heard about. Okay, so there's a lot to be proud of, obviously. So we made progress in a number of different areas, but still, I want to basically stress the point that we still need to work in some areas, particularly in terms of quality control. And so we are all in agreement that safety and functionality of cells are very important. We would all agree that reproducibility is very important. So one thing we are putting a lot of focus on at NCATS is actually trying to use robotic systems to automate and take out the human bias out of the process. At the same time, keeping very practical things in mind, like cost efficiency. We want to make sure that we have always a renewable source, and that it's actually a beautiful opportunity using iPS cells. We can generate cells in a patient and disease-specific fashion, and basically optimize all the processes. And it's also important to have cells that remain stable during manufacturing, so thinking very practically. So at the end, it really comes down to evidence-based regenerative medicine. And also, despite of making great progress in some areas, we still need to remind ourselves that we still have, in some areas, the limited understanding of the behavior of cells, and how can we go about this. And I think now with the ever-increasing advanced technologies, there's a great opportunity to generate these data sets, share this information across different laboratories, and also actually go deeper and then try a more comprehensive and integrated approach of better understanding why cells, for instance, different iPS cell lines have different growth rates. So why is that? Is that something that we can use in a predictive fashion to better understand cell behavior, eventually bringing functional cells into the clinic? But on the clinical side, again, quality control is extremely important, and based on what disease we want to treat, we have to keep in mind the disease features, for instance, how we want to design clinical trials. We heard about that. But also the timing of grafting. So I think the Parkinson's field has learned a lot about it's important to select the right patients and treat them at the right timing. And but at the end, you also have to think about, you know, how can we basically trace functionality over time and combine this with rehabilitation. So, so altogether, I mean, the comprehensiveness and working in a team science approach and making sure that many of the things we have learned, we also critically revisit. And I think this is a field that's moving in a very quick way, but we basically have to always keep in mind that now it's actually the opportunity, realizing that this is a turning point. Things that we want to do is, you know, can we really bring this into the clinical stage and keeping in mind that guided by the principle of not doing harm as an important component of this effort. So there will be a lot of more discussion about how to actually do this and can we work across different disciplines. And this is including basically automation engineers, biologists, chemists, folks who know about safety and toxicology, and move this great technology, in this case focusing on iPS cells, into reproducible therapy options. So I think I should stop here and I don't know if there are
Thank you.
time for questions.
Thank you. And now we'll move on to Jane Bowski, so is the Chief Scientific Officer of Asterias Biotherapeutics.
Yeah, thank you. There are many, many, many different scientific, technical, and operational challenges that face us in the cell therapy field, and I was trying to order them in my mind, and again, came up with several different questions. But I think one of the features, important features, is understanding what you're trying to have your cells do. Is it a neurotrophic effect? Is it a neuroprotective effect? Are you looking at producing a variety of factors? Are you looking at modulating the immune system? Are you looking at permanent engraftment and repair? And understanding what is you're trying to do with your particular cells, particularly in the context of the disease that you are trying to look at. So for instance, I've been in spinal cord injury for many, many years now, and know that if I'm looking at trying to repair the injury site in the context in the subacute injury versus the chronic injury, you're dealing with two completely different types of lesions that you're trying to repair. So I think it's really important that we understand what we're trying to do with our particular cells and what is that disease environment. Do we need to modify that disease environment? What is our therapeutic window for those particular cells? Again, it'll depend on the type of neurological diseases. I think another area that is extremely important is looking at how long do these cells need to be there, okay? Are they modulating, are they eliciting a permanent effect? What is or is it more of a transient effect? What do you desire? And how can you deliver the cells to the site of action? In many cases, we might need the cells need to be very focal. In other cases, might need it to be completely spread across the brain. So I think it's very important to look at how are we going to deliver the cells and get them, again, very efficiently to their site of action where they're needed. Do we have to look at multiple doses, and how do we modify multiple doses and multiple administrations and how do we measure that? What kinds of animal models do we look at it? I think one of the things that's been touched on here is the effects of the immune system on your cells, how resistant are your cells? Do you need to give immunosuppression if it's in the allogeneic setting? Even, and what are the effects of inflammation on your particular cells? I think one other area that is important is looking at, again, as Scott had indicated, which is manufacturing. And how can you manufacture your cells reproducibly? And what are the tolerances in each step of the way, okay? Can you put twofold more cells into this reactor? Does it have to be exactly 1.0 times 10 to the 6 cells per mil? I think the tolerances in your manufacturing is something that is very critically important. And I think one other area, again, that was mentioned is looking at cells and looking at critical assays for measuring not only the phenotype, the function, and the properties of your cells. So for instance, we, again, as was mentioned yesterday, we tend to look at the surface markers, various other secreted factors that can be produced by our cells. But we're only at the tip of the iceberg. And it was mentioned yesterday about big data. That gives me sometimes a headache because I see this enormous amount of data that we have on these cells, and I get frustrated because I don't know how to interpret it. And I think fundamental work into the basic cell biology and how these various parameters affect the functioning engraftment of your cells is very, 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 very critical. So again, a fundamental understanding of the cell biology and how all of these various parameters that we now have the luxury of being able to measure, how does that fit in to really understanding the safety and the activity of your cells? So I'll stop there.
Great, thanks, Jane. Now, scoot back to our FDA anchor. So now we have Thomas Finn, who is at the Cell Therapeutics Branch, Division of Cellular and Gene Therapies, Office of Tissue and Advanced Therapies, CBER FDA. Thanks, Scott.
Okay. So we heard two really great talks today, and also a lot of interesting discussions. And I think what's important to think about these two stem cell talks is you have two very different types of stem cells, but they were developed in a very different way with a different strategy, with a different target. And then they went back and looked, both preclinically and also looking at the field, what was learned in the field, about what would be the best strategy and the types of properties you would want to have for that particular product. And I think that's quite important in terms of deciding what characteristics, what specifications your product would need in order to do what you want it to do in the patient. And Jane mentioned you need to think ahead of time about what are you really trying to achieve in the patient. Do the cells need to migrate, in order to do they need to spread throughout the brain, or is it important for them to stay in place? And that's a property you could probably measure in an animal and in a dish, and it might be something that would be important. It's important to think about not only what you do want in a product, but perhaps what you don't want. So Lawrence talked about, the experience with fetal tissue transplants, and the question was raised about, well, how do you decide, what dose to use in your cells? So it was brought up, well, yes, it's great that, you can have a source of cells with their fetal tissue or what they're using, where you're making, purified midbrain dopaminergic neurons out of their stem cells. But that serotonergic cells might be bad. So not only do you want to make sure you have the right types of midbrain dopaminergic neurons, but perhaps make sure you don't have serotonergic. And you can think about, well, some of the patients benefited for that fetal tissue, and maybe the differences were that, maybe there was differences in how they were transplanted, maybe there were differences in patient populations, some benefited, some didn't, maybe that was the explanation. But there could also have been differences just in the quality of the fetal tissue. And 1 or 2 days difference in when you collect fetal tissue can really change the properties and ratios of the types of cells that were there. Another issue is just, can you get cells to survive after transplant? That's a big challenge in many different fields. And you can learn not only from, the neurologic area, but also other areas where you're trying to transplant cells. So there's common issues, and I think there's common opportunities to learn from other fields. You know, on the topic of, you know, getting cells to survive, one thing that's often measured, probably ubiquitously among all stem and gene cell therapies, is doing trypan blue staining. Okay, well, that's a good assay. It's cheap, it's simple, everybody can do it. But the common interpretation is like, well, that means my cells are healthy and happy. Well, it tells you you haven't killed your cells. It doesn't tell you that the cells are healthy and happy. Patients in an emergency room have a pulse. They're not necessarily healthy. So characterization is important. Also, it was mentioned and brought up in some of the talks and also some of the discussions about how you're going to deliver the cells. And also, are the cells stable, you know, in the emergency room as you're going to transplant them? And so we often ask, you know, to really think of not only, you know, what's the product quality at the GMP facility, but is this really compatible with your, you know, your clinical protocol? If you're going to have a neurosurgeon transplant these, what device are they going to use? Just because they're using a cleared device, well, it might be a cleared device for a small drug. Is it really biocompatible with your cellular product? And then, neurosurgeries don't happen in 15 minutes. You know, if the cells are sitting around in a long time in the surgery room, is your formulation, is how you're storing the cells, is how, you know, they sit in the injection device, is that really going to be compatible with your product? And then again, I would just emphasize the importance of kind of learning from other fields. So some of these things are unique to the neurosciences, but a lot of, I think there's a lot of opportunity for shared knowledge, especially in preclinical testing and bench testing.
Great, thanks, Tom. Okay, so what we'll do now is we're going to move on to key questions 2 through 5. And here what we'll do is we'll call out specific people to kind of comment on that. And I will say, if it's something that you felt you've already said, you know, you just use it a time to reinforce that and highlight it. Okay, so what we'll have for key question number 2, we'll have Sean and Jane comment on that. And go ahead.
Sorry, just making sure it's working. So I will, I know Jane will speak about maturation, differentiation, but I'll speak to, I think that to look at this question, it's very much going to depend upon the goal and the target overall. What's the intention overall? It makes sense to be thinking about a neurodegenerative disorder where there is a selective degeneration of dopaminergic neurons, and the goal is to undergo a cell transplantation approach. Also recognizing, of course, that there are other neurotransmitter systems that do degenerate over time, even in Parkinson's disease. But if we're talking about a completely different approach where we're looking at MSCs being injected, let's say, into the brain to be able to stimulate repair mechanisms in a patient chronically disabled by stroke, then perhaps the goal isn't really to be thinking about maturation or differentiation, that the cells are there to do a specific function, and whether they stay stabilized or they go away and die, you know, perhaps maybe even that may be preferable. So to answer this really does depend upon what the overall goal is. And you know, we think about various different neurological conditions where we'd like to be able to come up with a tissue replacement strategy. It gets more complicated to think about other conditions when we go beyond Parkinson's disease to other neurodegenerative disorders. You'll have to require a completely different thinking process behind if we're going to put in cells to the brain to try to think about taking on Alzheimer's disease, for example, where you have so many different cell types that get damaged over time, where here, you know, what we're talking about may be a precursor cell where it would have to differentiate into a lot of different cell types, for example. And that's why I think in many ways Alzheimer's is probably not very high on the list compared to Parkinson's disease overall for this kind of strategy. The other comment I just wanted to make was really, when we talk about physiologic function, how to optimize it. And while I think we've had some good discussion already about that, it is all about trying to understand mechanistic targets. And so what I'm hoping is to try to think about optimizing function is going to be doing these types of studies where we build in mechanistic studies. The work that Athersys is looking at building in spleen for instance, volumetric analyses or doing white matter tract analyses is going to be, I think, helpful to be able to understand, those patients that actually have improvement on those endpoints, then that will be the target to try to focus on to optimize the effect. And from the perspective of looking at mechanism, how can we link that back to trying to find appropriate in vitro potency assays so that we would have assays that would tell us in the laboratory that these cells are really good at their intended effect that's predictive of what they're going to do in the human brain. I think that's where we need to go to try to optimize effects. And then the last thing I'll just bring out is that many people have brought this up, that it could very well be that a single injection may then need to be followed by another injection, and perhaps that's a different delivery route. So the intravenous route might be appropriate to take on the inflammatory response after an acute neurological injury, but then maybe perhaps that needs to be followed later by an intracranial injection later because the immune targets are gone, but the cell therapy may still have a chance to work directly on the brain.
Great, thanks, John. Jane?
Yeah, I really don't have that much more to add to that. It all depends. The optimal stage of differentiation or properties of your cells depends on what you're intending to do with those cells and what you think that their mechanism of action is going to be, whether it again be neuroprotective, whether it be paracrine, whether it be immunomodulatory, whether it be integrative and reparative. So again, you want to look to see where do your cells need to go, where do they need to migrate within the tissue that you're targeting, how long do they need to stay alive, and what do they need to do when they're at the target tissue. So I think, again, a lot of fundamental work that needs to go into an understanding, how your cells are working, how they might interface with the macroenvironment and the microenvironment of the injury site or wherever they need to go. So again, I think it's really fundamental that understanding what the cells need to do for the disease and disease stage that you're looking at treating.
Great, thanks, Jane. So now for the second part, question 2, we'll have Sue Chun comment.
I think that it actually is linked to, this question is actually linked to what cells, what stage of cells we are going to use. Because again, it also depends on what we want to achieve. I mean, I'm more interested in the area, for example, we want to reconnect the neural circuits in order to achieve a relatively permanent effect, like Lorenz talked about. And in that case, we may have to use very specialized cells. And I mentioned briefly that we may have to use cells at a particular stage, because at this immature or progenitor stage, these cells have a robust growth capacity. And they, for whatever reason, they actually knows where to grow and to find their target and form this functional connection. And if that happens, then the functional outcome will be relatively stabilized. And another aspect of the nervous system is that kind of activity dependent. Besides physical cell transplantation, I think this stimulation, many different kinds of stimulation, let's say rehabilitation kind of strategy may also be extremely useful to promote, stabilize, or sustain the functional outcome. So I think there is a branch called regenerative rehabilitation or something like that is ongoing internationally. In terms of immunological tolerance, I think this is probably general to all the cell therapy field. The nervous system may be slightly less concerned if we can do a short-term immunosuppression, like 1 year or something like that, based on previous studies. with fetal cell transplantation, for example. But I think the idea might be, use autologous transplantation like we actually did with the non-human primates, and you probably don't need immunosuppression. Of course, it comes with the price in terms of time, cost, all sorts of things. And there are many other ways for dealing with immunological tolerance. I think that these kind of things have been, I think, was discussed yesterday, and so I'm not going to continue. I think that the tracking system really need probably some newer development. I think for Parkinson's disease, for many of the neurotransmitter release or something, we use PET. I think Sean mentioned that need to develop imaging system for monitoring microglia or macrophage, where they are, what they are doing. I think this will really help following the transplanted cells, where they are, how they are doing actually, and to combine with the end outcome measure, and that really will help the cell therapy field.
Great, thanks, Chen. So now Scott will move on to question 3 and get back to product development challenges.
Thank you. As we think about scaling up, typically here we're talking about allogeneic or quote-unquote universal donor products. One of our big challenges is comparability. Actually, comparability is a big challenge nearly always, but never more so than with scaling up, because typically here we're moving from one kind of cell expansion technology, perhaps culture in tea flasks or relatively small-scale suspension culture, to something much, much bigger that usually requires a dramatic change in the expansion technology itself. To get to really big scales of cell expansion, typically we're going to have to use something like a stirred tank bioreactor, culturing adherent cells not on flat surfaces but on microcarriers. And this is, from the cell's point of view, a pretty dramatic change. So comparability is very much an issue there, particularly for those cell types that require a lot of crosstalk between one another. Growing these cells on particles that are floating around in a suspension system is going to be a very different environment for cell-cell communications and so forth. Hollow fiber bioreactor technology, to some extent, can be used here, but comparability is a major concern there. Let's see. With scale out, typically here we're talking about patient-specific products, typically autologous products. And here, closed system single-use technology, although it's valuable for scale up, it's absolutely crucial for scaling out, because we're scaling out typically on throughput, on parallel processing multiple products side by side. We're only beginning to actually execute on that strategy. But clearly that's the way that one is going to have to manufacture large numbers of patient-specific products, autologous products, or in some rare cases, matched products. That takes single-use closed system process technology and really good automation. Fortunately, by now, there are closed system, single-use disposable process technologies and semi-automated process technologies for carrying out nearly all of the unit operations necessary to manufacture cells. Nearly all. One big glaring hole is the kind of processing that is typically, or that sometimes needs to be done at the clinical site pre-administration. And this has been touched on just a little bit yesterday and already this morning. This is often overlooked. It's very easy to think of manufacturing as being something that we do within the manufacturing GMP facility, and that's it. However, in many cases, the product might be shipped cryopreserved to the clinical site where it needs to be thawed and, heaven forbid, washed and resuspended into a medium that is compatible with administration to the clinical site. Now we're talking about doing a manufacturing step completely outside the control of the GMP facility, and worse yet, something that eventually will need to be done not just at a major academic clinical center that's participating in a clinical trial, but eventually that needs to be done at a hospital in Spearfish, South Dakota. Apologies to anybody who's from that town, by the way. It's just such a great name, and it's a perfect example. Developing automated or semi-automated process technology for final processing, for thaw, resuspension, washing at the clinical site, and doing that in something that's a small enough package that you can actually, and that's heavily enough automated that you can put it on a bench top at the clinical site. One other point that I think is worth mentioning about scaling out is that as we're doing process development and as we're building our understanding of critical quality attributes and so forth, when we're scaling out, we have to bear in mind that we're scaling out on patient-derived raw material. We're doing parallel processing with cells that are coming from patients who, by and large, are sick people. And the thing about getting cells from sick people, patients, is that you'll have a much higher degree of variability than with healthy donors. Furthermore, you have to take into consideration pre-treatment effects. We saw this a lot in cellular immunotherapy, where patients who were getting immunosuppressants or steroids who've gotten cyclosporin 6 months before the cells were collected give very, very different results than patients who didn't get steroid treatment in the last 6 months. The same, I'm sure, is going to be true of cell therapy for neurologic applications. So that needs to be borne in mind as well. And last of all, if I haven't emphasized it enough already, and I can't emphasize it enough, the challenge of comparability. Because anytime we change anything about our manufacturing processes, whether we're scaling up or out, we have to worry a lot about that. PAT can help us, process analytical technologies can help a lot with that. And with that, I thank you very much.
Great, thanks, Scott. Okay, so we'll move on now to question 4, addressing readiness in general. And here we'll have Sean, Elias, and Jane comment.
Okay, yeah. Just want to reiterate again that, working in the team science environment, it's I think going to be very critical. And so as an example, so similar to the cancer field, where patients are diagnosed and treated in comprehensive cancer centers, I think we should also think about moving into an stage where we think about comprehensive regenerative medicine centers, where basically teams can work on exactly the important things we're talking about here today. So engineers, basic biologists, stem cell biologists, and also clinicians. And I think it's going to be very critical here to maintain a high standard. I mean, first introduce standards. That's something that we also need to remind ourselves. So standards is something that will always come back as a bottleneck. And so just looking at stem cells, what exactly are standards, and why are variability endpoints that we detect? So how can we better understand and control those variability issues, for instance? So this all feeds into each other, and without having this comprehensive approach and thinking early on the preclinical stage, how we can, for instance, generate protocols that are GMP compatible. So it doesn't need to be GMP compatible at the beginning, but if you can incorporate that into your thinking early on, I think this will help us to to make quick progress once we try to translate the technologies into the clinic. And so this is going to be key, I think, not only for pluripotent stem cells, but for all the other cell types and sources that we have heard of over the last day. So it's going to be critical that we also share information in a different way than we have been doing this in the past. For instance, that we nowadays you can quickly upload information and share and use systems biology type of approaches to actually better understand, trying to better understand the biology, doing very basic things sometimes that is missing, oftentimes in protocols, dose response, the effect of using combinatorial factors, how does this really affect cells? And I think we have to keep, reminding ourselves that rigor and reproducibility is something that will basically hunt us if we don't make really decisions now to really take this on. Yeah.
Thanks. I think all of these factors are important. The critical quality attributes, process how you make your cells, and materials you use for potentially making your cells and administering your cells. It's all a balancing act. I mean, you can't strive for perfection and then start testing it in the clinic because you might not ever get there. And it's really important to understand your cells and how they relate to the disease environment. And sometimes animal models don't appropriately predict what you're going to be seeing in the human clinical environment. So I think it's really a balance of doing the best you can to understand what are the critical quality attributes and understanding your manufacturing processes. How can you potentially make your cells and make them in a reproducible way, but knowing full well that you might need to come back and review some of those critical attributes and your processes and your materials that you're using as you get further and further and further in the clinic.
Thanks, Jane. Sean?
I'll just add that I think that Elias really did quite a nice review based upon these items in his talk. Just on that, for the neurological space, if there is a relevant animal model, that I think many of us feel that it's appropriate that that's where all of this starts, that there would be data before you're going to go forward to a clinical trial, that the cell therapy product be tested in a relevant animal model for safety and for the intended effect, whatever that effect may be, since we've talked about that there's a number of different possible mechanisms. And that reproducibility, as was mentioned, I think is also important, that there is effect that's seen across laboratories, for example, or other groups are finding the same effect. But then, when a cell therapy product actually does then go into clinical trial and it's very well characterized, it's been shown to be very safe, and it's already being shown to have biological effects in certain patient populations, I do think it's appropriate to have discussion about, whether it might be appropriate for another patient population that makes sense biologically, and that one doesn't have to go through an extensive series of preclinical animal studies over again if there already is a lot of known information about the cell therapy product in the patient populations that are relevant. And so in this respect, I think we could probably move the field forward if we don't require an extensive amount of preclinical studies if you're thinking about extending to another patient population under those conditions.
Great. Thanks, Sean. Okay, so we'll move to our final question, and I think what we'll have is Tom comment on that as it's the key regulatory challenges in the next 1, 3, or 5 years.
Okay, all right, great. I'm encouraged that, Jane, started out her answer by saying it depends, because that's a classic FDA answer. So I think it's really interesting to watch how this field has moved forward in the last 10 years, especially the last 5 years. And a lot of the people around this table have been doing this for a lot longer than that. There's also, just a lot of experience out there in the manufacturing sector. And one might think that, a lot of these kind of manufacturing challenges are very new. These are advanced therapies. And so, you're going to have to start from scratch. But actually, there's a lot of, now big pharmaceutical companies, even small molecule companies that are interested in getting into this field. And a lot of their experience does translate over. But there are still unique issues. When you talk to process engineers in other fields, that are normally making monoclonals, you can just see the gears turning in their heads as you're describing some of the unique challenges and some of the things you have to do. I would reiterate that it is really important to learn, from studies done on the bench, studies done in animals, how might this product work? Is it really working the way you think it is? I mean, sometimes some of the best properties about, stem cells and other types of cells is also some of their, biggest caveat in terms of manufacturing. So, for example, you have a stem cell that can do many different things. Well, that's great, but then you go to, your engineering team and say, okay, now come up with specs. Okay, well, how does it work? Well, I don't know exactly how it works. It could do this, or it could do that, or it could do something totally different. And maybe in the different patients or different patient populations, it'll do different things. Or maybe, for, stroke, it'll do this, and spinal cord injury, it'll do that. That can be very challenging to say, okay, well, now this is, what I'm going to, optimize my manufacturing. I'm going to do certain unit operations to select for this population or drive that, or make sure at the end I've got this. And that I can say, okay, this is the right cutoff to say, yes, this is good enough to use in a patient, or no, we have to start over. So that's a challenge, and so learning, having good preclinical animal models, doing the proper characterization work is quite important. Also, it's useful, and it was brought up today that, you can, it's not just like, well, you get your phase 1 trial off the ground and then you could forget about manufacturing other than just producing the product. Product characterization doesn't stop there. FDA, we always talk about the life cycle approach. And, from the CMC side, we don't just think of a clinical trial as collecting safety and efficacy data. Obviously, that's one of the big points. But it's not just about the clinical and statistical data. It's about learning from your manufacturing. What does that maybe tell you about how your product is working, or what's important, or what you need to fine-tune? And I think there's opportunities for like biomarkers, clinical outcome measures. Robert Mays talked about, well, in some of their preclinical studies and some of their outcome measures are looking at things like, increases in Tregs or decreases or shifts in cytokines or different T cell populations. That can be informative. You can also be informative, asking the question, okay, well, I'm going to immunosuppress for 12 months. Well, why 12 months? If the cells don't survive longer than 6 months, does it make sense to still be immunosuppressing for 12 months? Is there a way to measure, neuroinflammation or possible rejection of the cells, or methods to make sure that the cells are still there, or methods to make sure that the cells have spread and migrated appropriately? So I think there's challenges going ahead on kind of the clinical side, looking at outcome measures, trying to match that up with product properties. I think there's also challenges, preclinically, trying to really make sure what's the best product to make and how to set those specs.
Great, thanks, Tom. So I think what we'll do now is we'll move to opening up the floor for questions for everyone. We have about 10 minutes. So if you come up to the mic, give your name and affiliation and ask away.
Good morning, Rosemary Hunsiker, NIBIB, NIH. So I think we all know that there's a really big difference from going from a T75 flask up to a single dose of what is usually inoculum, which is like 10 to the 9th cells, something like that. And those challenges have been discussed to some extent here. And in the neuro field, I think it's probably even more prescient than it is in maybe some other application fields. But the one thing that seems like a big challenge here is that We have a lot of exciting academicians coming up with really, really cool, interesting, wonderful theories and getting at mechanisms. And then there's a lot of really serious people looking at how do we do that scale up just from a purely brute force kind of way. But there's not a whole lot of people in the middle that are forcing that kind of communication to happen. But there's real science and engineering challenges there. I mean, this is a place where you could, you know, dare I say it, actually even write a grant. So I think the onus is on us at NIH to find a way to give you guys a more comfortable place to write such grants and to have them reviewed without being laughed at. So that's on us, but what's on you is to have those communications and make that happen. So I'm wondering if you guys wouldn't be willing to entertain at least a few ideas out here in this room about how we might do that from that practical point of view, is to force those connections and communications to happen more robustly.
Does anyone want to jump in? I mean, essentially, it was touching on some points, I think, that Ilias was making.
I mean, that's absolutely right. So we have to just think really outside of the box and then maybe go really also outside of our comfort zone, right? So to reach out and interact with people and simply realize from the very beginning this is impossible just if one little group is trying to solve these big questions by themselves. So this is impossible. So we have to realize at some point, you know, if you want to scale up and you simply don't have the adequate technology, so you have to go out and interact and collaborate with somebody who has that experience. It's still for us very humbling to see that, you know, very simple questions of standardization. So if you take a dozen of different pluripotent stem cells, you plate them, you see some grow faster than others. So why is that? Right? So it's what does this? I mean, there's some biology behind this, obviously, but so if you don't have a technology to do, for instance, 24/7 imaging, you would miss such important information and to define what's actually what's the range of normal, and if there's an outlier early on, and you could maybe early on just simply on that you know, information say, look, the cell is probably not you know, going to perform adequately in your differentiation protocols, for instance. So we still have to realize that simple differentiation protocols are very difficult to reproduce across different laboratories. Why is that? Maybe we should just you know, do more site visits or have meetings where, you know, hands-on training is possible, you know, video tutorials. And that's actually something we are trying to do at NCAT, so supported generously by the NIH Common Fund. So we're taking such questions really head on and trying to do this also in a highly collaborative fashion. So, so we will collaborate through strong collaborative networks here intramurally as well as with academic groups and biotech companies. So it really is still a huge challenge, and everyone agrees this is an inflection point, but now to get to the next level, so we have to really work across borders and realize that's impossible. And so Lawrence's talk was giving us an idea about all the complexities of how to bring this technology into the clinic, and even questions about patient recruitment, open label, or, you know, double blind, and all those kind of things have been historically discussed, and I think so has not been resolved, and so this would be the opportunity to go after those kind of questions.
May I add a little bit on the technical side, how to scale up the cells? Because I mean, we can use brute force to have a huge starting material like stem cell, iPS or ES, but there are many other ways. One way is when we differentiate the cell to a committed progenitor cells, let's say blood stem cell or dopaminergic neuroprogenitor cell. The question is whether we can actually expand this population without changing their fate yet you end up with a uniform population. And this is actually scientifically very challenging, but people are working on that, and that probably requires collaboration between bioengineering and biologists. And actually, we are working on similar things. And I think NIH could potentially support this type of effort, because in the end, that kind of product will actually help the cell therapy field, because if the population is reproducible, predictable, and for production, it's easier, and I think it's actually a pretty good way to promote that direction.
Thank you.
So I hope that people think about this as an opportunity to actually write grants, to take on the challenge of, sometimes you have to fall on your sword a little bit in the beginning, because study section needs one or two to look at first, and they're going to get triaged. But if you keep hitting the nail, you might get there. And finally, I would just say I'm not in any way, shape, or form involved with the new Manufacturing Innovation Institutes, but that looks like a wonderful platform for people to be able to come together, and I would hope that everybody just sort of checks them out and see if that's a way that we could take all of these stakeholders and pour them into one pot and see what comes out.
Okay, thank you. Go ahead.
Nadia Lomelski, NIH. I want to raise a question about manufacturing, kind of building on what Rosemary said. So what this panel to comment on is whether we are ready for a centralized manufacturing, where given the complexity of the field and so many specifics for each applications that exist at this time, and need to monitor very closely what happens with each batch of the cells being manufactured. Is it preferable to have a type of facility that Lawrence described, which is in Sloan Kettering, and they have day-to-day communication with the manufacturer, or are we ready for a large facility where cell orders will come from different universities, companies, geographically removed from that facility, and where expertise would be, again, geographically removed? I can see the advantage of centralized facility, and I'm not going to name all those advantages. The question is when? When is the right time to go to centralized facility? Are we ready for that? Or we're still at the stage where the sponsor needs to be very closely in communication with the manufacturer, which is difficult to do when centralized facilities.
Who would like to tackle big or small?
Well, there's a company right up the road that is our contract manufacturing organization called Lonza that does that, and they've been doing it for quite some time. And I think that the field, the cell therapy field, and again, maybe we haven't grasped it here in the United States as firmly as potentially the Japanese have, but the Japanese, every one of the big corporate entities in Japan has started to place bets on cellular manufacturing for the field of regenerative medicine. We only have Lonza, to the best of my knowledge. There are other smaller, I don't want to call them mom and pop stores, but there are more cellular manufacturing organizations coming up and trying to compete. But Lonza has the greater majority of the share of the cell therapy field, to the best of my knowledge. Different individual companies are trying to take it on themselves, but it's an economy of scale, and if you're a smaller company, you can't do this in a trivial fashion.
Okay.
Maybe just very brief, because we obviously try to think about that question quite a bit, and I think I would say the FDA response, it depends. No, so it really depends in which cycle of product development you are. I mean, for us, it was very, very nice to have actually the GMP facility right next to it, because the problem was not really necessarily our lab or people that have experience in running the GMP facility, but kind of the people in between. So it took quite a bit to actually find someone who can translate the language very well. In our case, it was actually this Mark Tomishima ran a stem cell facility before, who thought a lot about process development, but he was not really a GMP expert. But so for us, it was very, very important to have this close interaction. And so obviously at some stage, when you maybe a little bit further down the road, or if you have such large facilities have experience on every single topic, then that might work. But I think there's still quite a lot of knowledge from the product itself that you need to know. And you need to have someone to translate that to a larger facility that they really can integrate in that process.
Thank you. Could I just follow up on one thing? Yes. So part of the equation of whether to have centralized or regional facilities depends partially on the product and what type it is. And not every product can be frozen. And if your product only has a 6-hour shelf life, well, that kind of takes a centralized approach out of the equation. The other thing to keep in mind, and this was brought up today, was that regardless of whether you're using centralized or facilities closely associated or even at the same institution, there's a lot of academic centers that now have their own GMP facilities. Is that because these are more often patient-specific, there has to be very close tracking between how the cells are collected from the patient, manufactured, and then given back to the same patient. So the traditional idea of there's the walls of the GMP facility, and then that defines GMP, kind of gets broken down. And sometimes it's a fuzzy line between practice of medicine and actual GMPs.
Thank you. Now, unfortunately, we're out of time. I'm going to let Scott make the final comment. I'm sorry, but maybe you guys can track these folks down at the break or throughout the day if you have questions.
Well, just very briefly, I very much agree. It depends on the nature of the products, and it depends on whether it's a patient-specific and autologous product or whether it's a quote-unquote universal donor product. I agree, Lonza certainly has a large market share for allogeneic universal donor mesenchymal stem cell products. There are a number of other contract manufacturers that have particular strengths in some other cell types and with autologous products. Ultimately, I think what we need is a national resource, if you will, not so much for the contract manufacturing, because that's what industry will do, but for the translational development, for everything that's going to lead up to commercial contract manufacturing, or that's going to lead up to, for certain therapies, distributed manufacturing, or perhaps something closer to point-of-care manufacturing for simple processes that can be heavily automated. We're beginning to see something like that. There's CCRM in Canada that's beginning to attempt to do that. That's something I think that NIH can be incredibly valuable in promoting. And perhaps it's not a single center, perhaps it's multiple centers. This is a little bit of what PACT was set up to do, but PACT tends to focus on very early stage clinical development. Something that goes beyond that and that involves the automation industry and that brings pharma in, I think would be a very powerful tool for advancing the field. So I'll put in a plea for that and hope, with hopes that NIH can foster that.
Great, thank you. And with that, that will wrap up the neurology session. I want to thank everyone, our speakers, our panelists, and I'll hand it over to Gary.
Terrific panel. You have worked so hard, you deserve a break. And we will reconvene at the top of the hour. Enjoy.